So we're going to finish up the amino acid lecture by looking at the last three types of structures that we can see with proteins, and that is secondary, tertiary, and quaternary. We've spent quite some time now on the primary sequence and the primary structure of a protein. This is basically the protein that's been denatured, and we've kind of unraveled it, unwrapped it into a straight chain amino acid, and then we've taken a look at what amino acids are included and the order of which they are showing up. So that is the primary sequence or the primary structure. How are the amino acids linked? Secondary, tertiary, and quaternary we have mentioned before. Secondary structure, we focus mainly on alpha helix and beta pleated sheets, and we'll see a picture of those in a minute. Tertiary structure is the 3D arrangement of all of the atoms in the protein. So how do they really take shape in a three-dimensional type of form? And then quaternary structure, most proteins have more than one unit. So how are these globs basically behaving with each other? So when they do come in together, what do they look like? What kind of shape are they taking? And we can study them and do the primary structure and everything else that comes along with it. But that is the quaternary structure of a protein. Like we've said before, we've mentioned already a primary, and we've mentioned secondary as well. And secondary, we said, were alpha helixes and beta pleated sheets. What you're seeing on the screen right now is basically an alpha helix. You've got to think of these things as slinkies. And when you take a look at the alpha helix, you've got to imagine a tunnel. And the tunnel on the inside is kind of barren. There's nothing really there. And on the outside of this tunnel, or the slinky, however, or the spring, however you want to view the alpha helix, on the outside of the slinky or the corkscrew, that is where the R groups are going to be located. And that's what this slide is basically showing you here. What is keeping this alpha helix together into this coil shape? Well, those are basically hydrogen bonds. And hydrogen bonds, of course, are going to show up in the amino acids because think about it. You've got a hydrogen attached to an N over F. And anytime we said that we have a hydrogen attached to an N over F, then that can associate itself with other hydrogens that are also bounded to an N over F, and those two can basically form an intramolecular or intermolecular bond, and that's the glue that's holding this alpha helix together for us. So hydrogen bonding, you can kind of see that's here. The blue, of course, are the nitrogens. The red are the oxygens. So you're actually seeing the amino group and the carbonyl group behaving together in such a way that it creates this hydrogen bond that's keeping this corkscrew nature in place. When the alpha helix begins to form, you can see the alpha helix here. All of the R groups are going to be sticking out of the outside. So this is the R group of one amino acid, this is an R group of another amino acid, this is an R group of another amino acid, and all the way down the line. So all of the R groups kind of point on the outside for the most part, and that's the typical kind of representation of an alpha helix. So on a homework or test or quiz, you're going to be asked to draw an alpha helix. Give me some information about an alpha helix so that I know that you've watched the video and that I know that you've read a little bit about alpha helix in the actual chapter and that you've listened to what I've had to say. So alpha helix, it's a coil. It stays together because of hydrogen bonds, and the R groups of the amino acids point outward on the helix surface itself. We also said that we have beta pleated sheets, and this is a representation of a beta pleated sheet. Imagine you taking a sheet of paper and folding it in such a way to make an accordion style sheet. That's what we've done here. This is basically a beta pleated sheet. So we've pleated the piece of paper and then we just kind of opened it up a little bit, and when we've done that, we get these kind of ridges or these valleys and these peaks. So the beta pleated sheet goes down and then it comes back up, and then it goes down and it comes back up, and then it goes down and it comes back up, and over and over and over. That's what we see. We see two different types of beta sheets that can happen. One we call parallel and the other we call anti-parallel. Let's take a look at the differences between them. It's just a very slight difference. If you don't look at the diagram closely, it's not really obvious of what's going on. For the parallel beta pleated sheet, you're seeing an amino acid chain, and the N terminus is up here at the top and the C terminus is down here at the bottom. And then this is another amino acid chain that's here. So the N terminus is up at the top and the C terminus is down here at the bottom. Notice that they go in the same direction, so N on both up at the top in this diagram and C at the bottom on this diagram. Because they flow in the same direction, we call that parallel. So this is a parallel beta sheet. And again, the beta sheets are held together by hydrogen bonds. Look at this. Here's the amino group, and then there's the oxygen of the carbonyl, and it's that hydrogen and that oxygen that's basically hooking up to make this hydrogen bond, which is the glue that keeps the beta sheet together. The other version is anti-parallel, and you can kind of obvious now what the difference is. N terminus is up at the top and C terminus is down here at the bottom. And then maybe what would happen is that a loop would go on and we see the N terminus here and a C terminus there. So they're running in opposite directions. However, if you look, the beta pleated sheet still has hydrogen bonding involved. It's still the hydrogen on the nitrogen and the oxygen of the carbonyl that holds it together. It's just really right across from each other in the anti-parallel beta sheet versus kind of diagonal off to a slant in a way in the parallel beta sheet. So those are the beta sheet stories. That's really all that we need to talk about secondary structure. So we've talked about alpha helix, and now we've talked about beta pleated sheets. What's next? Well, this is a tertiary structure. Not quite yet, though, but we're kind of building up this process or this idea of what a protein is supposed to look like. And we can take this secondary structure and we can look at the alpha helix and we can look at beta pleated sheets and we can start to make these 3D orientations of proteins that I have. And this is the protein for carboxypeptidase A. Remember, carboxypeptidase A, those are one of your XO peptidases, right? It focuses the end terminus or the C terminus of the primary amino acid sequence, and it cleaves that one amino acid off, and it tells you what amino acid is at the end. We often said that this was a protein, this was a protein, this was a protein. Well, this is basically the representation of carboxypeptidase A. It is a series of amino acids, and these amino acids have began to make some alpha helix that you're seeing in the diagram. The alpha helixes are purple, so you can kind of see the corkscrew. There's one here and one there and one here and one there and one here and one there. So they're everywhere. And then you take a look at the beta pleated sheets, and they represent these with the arrow, a flat arrow. So that's what you're seeing here, and then we see another flat arrow there, and one here, and one there. So this gives you some insight of what the 3D orientation of carboxypeptidase A would look like, and there is the representation of it. So when we begin to talk about tertiary structure, this is kind of what we're building up to, right? We're building up to what does this thing look like in a three-dimensional fashion, and then let's take a look at the alpha helix and the beta pleated sheets because it will give us an idea of how this thing begins to structure on itself. So there's the primary sequence. Understand that the tip and the end of this whole ravel of amino acids, we do have an N terminus and we have a C terminus, and this strand is made up of amino acids. And these amino acids are beginning to do corkscrews, and these amino acids are beginning to do beta pleated sheets. And because of that, the protein is beginning to fold. And when this protein begins to fold upon itself, it can take multiple shapes. This is the shape for carboxypeptidase A. If this shape is different, or if this shape changes in any form or fashion at all, we can completely disrupt the protein function, and the protein will not do the job that it's supposed to do. So this is one of the reasons that we saw issues with proline back in the primary protein sequence. And the reason for this is because these types of proteins are looking for specific shapes. And again, proline changes that shape at that location, and the protein cannot work because of it. So that's why, again, Edmonds cyanogen bromide worked. They weren't looking at shape, they were just looking at true basic chemistry. But if you look at these endos and exopeptidases, they're looking at proteins, they're looking at specific shapes. That's how they do their job, and proline disrupts that. So what other forces contribute to tertiary structure other than these hydrogen bonds? This is something that we've mentioned before, and this diagram just kind of lays out all of the bonding that can happen that holds the protein together in its shape. So if we start up here at the N-terminus, right here, NH3+, and we just kind of go along the choo-choo train of amino acids, well, here we see an amino acid with an OH group, and look, that OH group can participate in hydrogen bonding to a carbonyl. So the side chains of the amino acids are very important. Side chains can be glue that holds the protein together, and here we see a link or a bridge, and we get this nice kind of loop or corkscrew turn that begins to happen in the protein. Here we see another one. This is just another amino acid, and then this is another amino acid. So the side chains of the amino acids themselves can behave together. So again, we see a bridge that happens with the protein, more glue that holds it together, but this also creates a nice little loop for us in the protein structure itself. Finally, we go down to maybe a pleated sheet structure. Look at this. It goes down and then it comes back up. So this beta pleated sheet, there's hydrogen bonding that happens everywhere, and it's super glue really this time around that holds that protein together in that shape, and we get this nice little arm of the protein that happens. It's all due to hydrogen bonds. We finally get a nice little loop here, and then we begin to see the alpha helix that begins to take shape. Think of a roller coaster, right? You're on a roller coaster ride of amino acids. Again, that alpha helix is held together by more hydrogen bonds. What hydrogen bonds are there? Well, you can go back and take a look. It's the hydrogen bonds between the nitrogen and the carbonyl, right? Okay, so if we keep on going down the amino acid chain, here we see more bonding that begins to happen. If we look at this loop, this is an amino acid, there's your isopropyl group. This is an amino acid, there's your sec-butyl group. So it's isoleucine and valine that they're showing there in that picture. And those two groups are all aliphatic. They are all carbon and hydrogen. They don't really want to associate with anything that's polar. And we begin to see the protein fold on itself because of what we call hydrophobic interactions. So some of these proteins, they are nonpolar. They prefer to stay in a nonpolar environment, and they kind of cluster together when that begins to happen. So we see parts of the protein that are taking shape because of the amino acids, and they want to be exclusive, right? They only want you to have a ticket to get into the party. They don't want everybody involved. Well, if we've got hydrophobic interactions, we can have hydrophilic interactions as well. So some of these protein structures, they are polar. If you put them into an aqueous environment, those structures will want to kind of hang out in the polar environment as well. So hydrophobic and hydrophilic, water hating and water loving is really what that means. Bonds hold the protein together as well. Some of these R groups also are charged, right? So we do see these electrostatic attractions that begin to happen. So full-blown polar charges. These are basically structures or types of bonding that can come together to make the protein take the shape that it needs to take. And then finally, we can't forget about cysteine. Cysteine can hook up with another cysteine and make a disulfide bridge, and we call that a cysteine molecule at that point. So we're taking cysteine, we're making cysteine, and we're creating a disulfide bridge in the protein. And then finally, we're at our end of the journey with our C double bond OO, which is our C terminus end here. So we've ended up with an amino acid that's right here at the end. So there's the journey for the forces that contribute to a tertiary protein structure. That'll be a test question, right? You know that that's coming. So give me a list of things that are basically holding the protein together, and hopefully you can spit out a couple of these, at least 3 or 4. So hydrogen bonding, hydrophobic interactions, hydrophilic interactions, electrostatic attractions, and disulfide bridges. So there's a total of about 5, 6 if we want to stretch it, but at least know 3 of them and talk about them in detail, so that way you sound like you know what you're talking about. Uh, what else other than a tertiary bond that's going to be involved, or a tertiary structure? Finally, we get this kind of space filling diagram for quaternary structure, and that's what we're seeing here in this slide. This is the quaternary structure of heme, or hemoglobin. This is what transports iron throughout your system, and oxygen. So it's all based on iron, of course. That's why it's called heme. So this protein has iron on the inside, and it's a cage molecule, kind of, you can think of it that way. And this iron basically will pick up oxygen and it will transport it to various locations. Notice that they have kind of done 4 blocks or 4 blobs of hemoglobin that make up hemoglobin. So 4 blobs of protein. We see kind of one blob which is dark green up here at the top, and we see another blob that's kind of a lighter green in the back, and we see the same thing happen here with an orange, and then orange in the back that's a little bit cloudier. There's 4 basic proteins that are coming together to make this protein called hemoglobin. So sometimes we call them compartment A, B, C, D. Sometimes we call them protein 1, 2, 3, 4. It just really depends on how they want to label it. But this is the difference between quaternary structure. So tertiary structure, if you look at it, is just its simplistic form. This is the protein, this is the alpha helix, this is the beta pleated sheet. A quaternary structure, however, is a little bit more space filling. It's also how that one protein glob is interacting with other protein globs that are coming together to make the protein that we know in this case as hemoglobin. So it's all 4 of these compartments basically joining together that we're looking at a quaternary structure. So there's the difference between quaternary. These proteins do get very hairy, they get ugly, they get nasty, but that's the name of the game. We've also talked about how to denature proteins, how to take this protein of a quaternary structure and how do we get basically an unraveling of it so that we can start looking at the individual amino acids. We talked about a high acid and high heat environment that we can put the protein in. That doesn't go away. So the pH is very dependent, so we can acidify it, we can really break all of these glues and bonds that hold the molecule together. We can use things called detergents, so it's not uncommon to put maybe some soap or some type of detergent-related compound in this environment, and we can kind of help neutralize some of those charges and glue that holds the protein together that way. Organic solvents are also very common. I can put an organic solvent in, and this basically will promote some of the internal amino acids to come out and play, and when that begins to happen, we start to see the protein unravel. Heat and agitation, of course. We can put this protein on a little bit of heat, get it hot and warm, and it will break all of the bonding up, the intermolecular forces in that protein. Thank you, General Chemistry 112, for teaching me that lesson. And then it can unravel and we can expose some of this primary sequence of amino acid for us. If you look up at the top, urea is also something very common, and what happens with urea is that they are able to kind of help promote some of these hydrogen bonds to break and open up a little bit because it is traditionally a salt. So urea is another one of those kind of compounds that we can put in. It's an organic compound, but there's some charges on it at the same time. Not truly a salt. I guess I misspoke, but urea is a compound that we can use to disrupt the hydrogen bonding in the protein itself. So that's really all that we need to talk about as far as the amino acids are concerned. You're going to look at the PowerPoint and you're going to see some of these diagrams that we have kind of flipped through this whole lecture series, maybe a little bit more detail, a little bit better than what I could have drawn them. But that's what you're going to see in the actual PowerPoint itself. You're going to see some reactions in there. You're going to see some mechanisms in there. Remember, it's not really up to us to talk about mechanism here like in this slide. This is not something that I'm going to ask. I just really want to know what is it and what does it do. You know, the theory is going to be as simplistic as it can be. We're not going to focus too much on mechanisms and reactions and that type of thing. But, you know, overall general idea of an amino acid sequence, a peptide bond, making proteins, how we can analyze those proteins, and some of the reactions that do take place with how to make an amino acid that we might want to use. Notice I didn't go into too much detail with the titration, but we can titrate amino acids as well. They have pI values. We talked about how to calculate the pI value, and you do that by looking at the pKa's. Well, a pKa basically is the flat area on a titration curve. So the first pKa can be here, the second pKa can be up here, and then the vertical line that happens between those two pKa's is the pI point, and that kind of goes with the equivalence point. So we can associate that to a titration curve, and we can titrate them, and we can measure the pH as this titration begins to change, and we can plot that data on a Excel spreadsheet and get these beautiful titration curves that happen because of the amino acids. This is the titration curve of histidine. It's got one of those R groups that has a pKa associated with it as well. So here's the first pKa, here's the second pKa, flat areas, right? The connector between the two, the vertical line, well, this is kind of an equivalence point. Well, the histidine has a third pKa value, another flat line up here, and then another vertical point that connects the two together, and there's going to be the true pI value because these two pKa's apparently were the closest together. So I would use that to calculate the pI value, and that's going to be another kind of equivalence point on a titration curve. So I can relate this to titration. It's not nothing that we do too much of anyway, but I can at least mention it, and you know that it's out there, and you know that it's a possibility. So we are done with amino acids. There's really nothing left to talk about. This is it of the amino acids and the proteins. Again, remember these structures. It's going to be very important on the test. And then second, you need to go through and you need to take a look at the primary sequencing examples. So what happens if I do trypsin, chymotrypsin on all of those? How can I get and build this protein from scratch from where I started with? So that's what I need you to take a look at. That's what you need to study the most of. And then all of the other things can be fillers when you get the time. But if I'm going to focus on a couple of things for the test, it needs to be structures, 3-letter and 1-letter abbreviations, and then you need to immediately go to those example problems about the primary protein sequencing. So congratulations, you are finished with Lecture 2, Proteins and Amino Acids. That's all there is to say. Again, the next lecture module will focus on lipids, and we will do the same thing. We will go through structure, we will go through identification, we'll go through a little bit of reaction, and then we'll go into more basically structure type of properties like we did with proteins, amino acids, and like we did with carbohydrates. So you are an expert, you are a pro. Now go eat your tuna and go eat your chicken, because all of that protein is going to be swimming around, and it's going to be happy when it goes into your tummy, and then your body's going to be happy when it begins to break down the amino acids, and that way it can store them back and use them for when you need them. So good food. Yum yum.